So I will just open the mobile application to keep up with your questions. Here we go. Okay, so let's start with the ocular embryology. Embryology can be sometimes a tough topic for many of us because it is like a story. You need to know where or when did the thing starts. One of the common mistakes in embryological teaching is that we will start immediately with the ocular embryology without referral to the general embryology. So you don't know where did the eye start. So you just state it is, or you just hear that it starts in the third week by invagination from the forebrain, but you don't know what is the forebrain and where did it come from and what are the neural crystals and how did they develop. So let's start first with the first 10 or 15 minutes about just a glimpse about the general embryology and the development of the three main layers of the ectoderm and the mesoderm and the endoderm, so that you will be able to know where are or what are the derivatives of each layer and how did the story begins from the first place. So starting from the very beginning, we have an ovum that undergoes fertilization and undergoes series of cleavage and divisions, first by meiosis, then by mitosis, until they form the 16 cell stage, which is called the morula at the fourth day, then the blastocyst, which is a single cavity mass by the fifth day, and then this blastocyst will implant itself in the uterine cavity by the 6 days of fertilization. So here are the early embryonic stages. We have fertilized ovum that undergoes meiosis and mitosis to form the zygote. And the zygote undergoes mitosis to form the 2 cell stage, 4 cell stage, and 16 cell stage or morula. Then the morula will transform into a blastocyst. This blastocyst will consist of an inner cell mass and an outer cell mass, or a cavity. So this blastocyst in the fifth day will be ready for implantation in the uterine cavity. So here is the journey that the ovum will take from the ovary. After the fertilized ovum will pass from the ovary to the fallopian tube and undergo a series of division until they implant itself in the uterine cavity as a structure called blastocyst in the sixth day. So we have now the blastocyst. The blastocyst is sometimes referred as a bilaminar embryonic disc. At this layer or at this time, the blastocyst will be formed of two cellular masses. The inner mass will be called the embryoblast, which will form the embryo or will form the developing fetus, and the outer mass will form the trophoblast, and the trophoblast will form the placenta. So the embryoblast will later on undergoes division into two layers. So this embryoblast will divide into the epiblast and the hypoblast. So as we can see here, this is the bilaminar disc or the blastocyst, which has an inner cell mass or the embryoblast and the trophoblast, which will form the placenta. So this inner mass or the inner cell mass will later on develop the two layers. One is called the epiblast here, outer layer, and more inner layer called hypoblast. So this is like a section, a cross section, and this is a longitudinal section showing the epiblast, and underlying the epiblast there will be the hypoblast. So what will happen next? The next stage of embryology will call the gastrulation. the gastrulation that occurs in the early third week, meaning that the embryo will develop three germinal layers, as we know. So it begins first with the invagination of the epiblast to form what's called the primitive streak at 16 days. This primitive streak is just a narrow groove on the surface of the epiblast, and the cells from the epiblast start migrating toward the primitive streak. So here is the epiblast, and here is the hypoblast. Here is the epiblast, and you can see that there is a narrow groove appearing on the surface of the epiblast that is called the primitive streak. Here we can see the primitive streaks. So again, the gastrulation or the development of the three germinal layers start by appearance of a shallow groove over the surface of the epiblast. This groove, it is called the primitive streak. And here, and from this, and later on, we will have the distinctive three germinal layers. So the endoderm and the mesoderm and the ectoderm will develop according to the different stages of differentiation. So the cells of the primitive streak will invaginate the hypoblast inferiorly, forming the endoderm. And the cells of between the epiblast and the endoderm will form the mesoderm, while the remaining cells of epiblast will form the ectoderm. So here is the gastrulation. We have a cross section here in the developing embryo in the bilaminar disc. After development of the epiblast and the hypoblast and the primitive streak here, so we can see that here this is the bilaminar that will transform into a trilaminar, or an endoderm, ectoderm, and the mesoderm and the endoderm. So how this can occur? This will be that the cells present at the primitive streak will descend on the high and invaginate the hypoblast, and this will form the endoderm. Here is the primitive streak, and here are the cells invaginating the endoderm, forming the invaginating the hypoblast, forming the endoderm. Cells between the primary streak and the endoderm here will form the mesoderm, while the remaining cells of the epiblast here and there will form the ectoderm. So these are the main layers that will give rise to the different body structures overall. So here is the hypoblast that will form or the cells invaginating the hypoblast will form the endoderm, while the cells between the epiblast and the hypoblast will form the mesoderm. The remaining cells of the epiblast around the primitive streak will form the ectoderm. So we have now the trilaminar germ cell layer that will develop different ocular structures. So here are a list of the structures developed by the ectoderm, and here is the list of structures developed by mesoderm, that the ocular structures that are developed by each of ectoderm and mesoderm, which are the main layers that will form most of tissues of the eye. Apart from ectoderm and the mesoderm, we have another important structure that is called the neural tube. So the neural tube starts by formation at the 20 important structure that will form the development of the central nervous system. So the neural tube is developing from the that will give rise to the ocular primordia. By saying ocular primordia, we mean the primary eye, or the primitive eye, which is called sometimes the optic vesicle. So the neural tube is the source of development of the eye, or the ocular tube. And the progenitor mesenchymal cells and the neural tissue develops, and the mesenchymal cells we will discuss now. They are a group of cells formed from what's called the neural crest cells that we will see now. So the neural tube will be formed by proliferation of the surface ectodermal cells to form the neural crest cells, meaning that the ectoderm, we just know what is the ectoderm from the previous slide. So the cells of ectoderm around the Primitive streak will have a specific character of differentiation and proliferation, and they will form what's called neural crest cells. These neural crest cells will move medially to form a neural groove that will cause elevation of both sides of neuroectoderm to form the neural fold, and later on it will fuse to form the neural tube. So here is again this process. This is the gastrula formed of the three germinal cell layers. Cells around the primitive streak here, at the edges of the primitive streak here, they are called the neural crest cells. So the neural crest cells are surface ectodermal cells. These neural crest cells will undergo proliferation until the closure of this tube will form the neural tube. So they are proliferate until there will be invagination of the neural groove until it will close, forming the neural tube. So here we can see this is the surface ectoderm around the area of the primitive streak. So the surface cells, this is the surface ectoderm, and this will undergo proliferation, or the neural crest cells will undergo proliferation, which is the in the green color. So when they proliferate, they will push the neural plate inside, or push the neural plate inward, leading to invagination, what's called the neural groove. And by a further proliferation of neural crest cells, they will close together and will form the closure of the neural tube. So this neural crest cells will later on will form what's called the mesenchyme, or will transform into mesenchymal cells. So the mesenchymal cells are arranged around the neural tube. So here we have the neural plate that will undergo proliferation and further invagination, forming the neural groove. Then the neural groove will close, forming the neural tube, and separated from the surface ectoderm, which will form the epidermis. And between the surface ectoderm and the neural tube there will be neural crest cells that will later form what's called mesenchymal cells. So the neural crest cells are the progenitor, or the primitive mesenchymal cells. So basically the surface ectoderm and the neural tube and the mesenchymal cells are the main structures that will form the developing eye. So let's have just more information about the neural crest cells. So the neural crest cells are a unique feature that's present in the vertebrate, like human being, that they develop from the neural ectoderm. So then the ectoderm at the area of neural tube, it is called the neuroectoderm. Neuroectoderm, okay. And between the neuroectoderm and the neural tube, there will be the neural crest cells that will later on assume some ectodermal and mesodermal characteristics to form what is called mesenchymal cells. This will share in the formation of different ocular structures, as we can see later, and the defect in this neural crest cells can lead to congenital anomalies that affect the development of the face and the teeth and vertebrae. So one of the famous syndromes associated with the damage or maldevelopment of neural crest cells will be the golden hair syndrome. So the golden hair syndrome present with hypophyseal or midfacial hypoplasia with limbal dermoid and preauricular skin tags and oral fistula and defect in the vertebrae. This occurs due to the maldevelopment of the neural crest cells. So let's see this nice video for some general information of what we have just mentioned of the development of the general embryology.
XX are paired in the female and XY in the male. Prenatal development is divided into 3 successive phases. The first phase spans the first 4 weeks of development. It involves cellular proliferation and migration with some differentiation of cell populations. The second phase spans the next 4 weeks. It is characterized by the differentiation of all major external and internal structures. This process is termed morphogenesis. The third phase extends from the end of the second phase to term. Further development is largely a matter of growth and maturation. The embryo is now called a fetus. The origin of tissues begins with fertilization of the newly appearing ovum from the adjacent ovaries. Fertilization then takes place in the distal part of the uterine tube when the sperm contacts the egg. The fertilized egg then grows and is termed the zygote. Progressive cell division results in a ball of cells referred to as the morula. The morula grows and begins migration medially to the uterus, which it reaches at the end of the first week. The uterine cavity, meanwhile, prepares for the arrival of the fertilized ovum. As fluid seeps into the morula, its cells realign themselves to form a fluid-filled hollow ball referred to as the blastocyst. Two populations of cells can now be distinguished within the blastocyst. Those that line the cavity, or the primary yolk sac, are called trophoblast cells, and a small cluster of cells within the cavity called the inner cell mass, or the embryoblast. It is the cells of the embryoblast that will now form the embryo proper. The trophoblast cells are associated with implantation of the embryo and then result in the formation of the placenta. About day 8 of gestation, the cells of the embryoblast differentiate into a 2-layered disc called the bilaminar germ disc. The dorsal cell layer is the ectodermal layer. These cells are columnar and they reorganize to form the amniotic cavity. This eventually becomes the epithelium or the epidermis. The ventral cell layer is referred to as the endodermal layer. These cells are cuboidal and form the roof of a second cavity called the secondary yolk sac. During the first 2 weeks, the axis of the embryo is established. It is represented by a slight enlargement of the ectodermal and endodermal cells at the rostral or the head region. Within the ectoderm, a structure called the primitive streak develops along the midline. This is a narrow groove with slightly bulging sides. The rostral end of the streak ends in a small depression called the primitive node or pit. At this node, the cells of the ectodermal layer divide and migrate between the ectoderm and the endoderm to form a solid column that pushes forward in the midline. This cord of cells is referred to as the notochord, and its function is to support the primitive embryo. On the dorsal surface of the embryonic disc, the ectoderm forms the neural plate, whose lateral boundaries elevate to form a neural tube. This will eventually become the brain and the spinal cord. The endodermal cells also form a tube, which will become the gastrointestinal tract. This tube elongates and anteriorly develops outpouchings that form the pharyngeal pouches. These cell migrations now result in complete separation.
Okay, so here is the part that concern us as an ophthalmologist. So we know now how did the neural tube form after separation from the surface ectoderm. And between the neural tube and the surface ectoderm, there will be what is called the neural crest cells that will develop the mesenchymal cells. So here is just a summary of the general embryology that is important for us to understand the ocular embryology. So let's start the ocular embryology. The formation of the eye vesicle or the ocular vesicle start in the third week by progression or by bulging of lateral part of the anterior portion of the neural tube. So as the neural tube is folding, it shows three dilatation. These dilatations are called the forebrain, the midbrain, and the hindbrain. So where does the eye comes from? The eye comes from the forebrain. So the forebrain will show lateral outpouching that they will grow laterally until they reach the surface ectoderm to develop the primitive eye. So let's see how does this occur. So here is the neural tube. The neural tube will show three dilatation like forebrain and midbrain and the hindbrain. Sometimes you are asked in the exam about the other name, which is prosencephalon, mesencephalon, and rhombinecephalon. So the eye will develop from the forebrain. So here you can see this is an outpouching of the forebrain. This is the forebrain. Okay, and here you can see that this is the optic vesicle. So they appear as an outpouching that migrates laterally from the forebrain, as you can see here. So they will develop from the forebrain as a vesicle or a swelling on both sides of the forebrain tube that start to migrate laterally until they reach the surface ectoderm. So the development of the optic vesicle is in close contact or in close relation with the development of the lens. So both of them depends on each other. So this depends on, or this starts by proliferation of the neuroectodermal cells of the neural tube of the forebrain. So these neuroectodermal cells will start to form the developing eye. So the neural tube of the forebrain grows laterally and form two globular structures. As we mentioned, they are called the primary optic vesicle or ocular primordia. And when this ocular primordia or when this optic vesicle touch the surface ectodermal cell, this will induce this ectodermal cells to proliferate, forming the lens. So the surface ectoderm in contact with the optic vesicle is the structure that is responsible for formation of the lens, and at this stage it is referred as lens placode or lens plate. So let's see how does this develop. Here we have again a section in the embryo. So here is the greenish layer, it is the surface ectoderm, and this is the neural tube. So here we have, this is the ectoderm, and here we have the mesoderm, and here we have the endoderm. So if we enlarge this section, we can see that this is the neural tube. At the area of the forebrain, they will undergo lateral pouching. So here we have a lateral pouch, and here is this another lateral pouch that will form the or this globular swelling will grow until it touches the surface ectoderm, like here, and this touching will induce a series of changes within this surface ectoderm to form what is called the lens placode or lens, the primary lens. By the end of the third week of gestation, we have three primordial structures. So we have the lens placode, as you can see here. This is the forebrain, and this is the outpouch of from the neural tube, which is forming the primary optic vesicle that grows to touch the surface ectoderm, which then this touch will form the lens placode. And this is the optic vesicle. And in between the optic vesicle and the lens placode will be the mesoderm that is surrounding the optic vesicle. And this mesoderm, in conjunction with the neural crest cells, both of them will form what's called the mesenchyme. And this is, here is the mesenchyme. So let's see this video about the early development of the eye to summarize what we have just mentioned.
Complex cellular interactions and genetic signals coordinate eye formation. During development, the different tissues of the eye arise from 4 embryonic tissue sources: ectoderm, mesoderm, neuroectoderm, and neural crest cells. The latter 2 tissue types form during the process of neurulation. Beginning at the dorsal midline, inductive signals from the underlying mesoderm convert ectoderm to neural tissue.
So there will be a specific area of the surface ectoderm that will develop into neuroectoderm,
resulting in the formation of the neuroectoderm.
So here is the neuroectoderm, and here is the neural crest cells that will undergo proliferation and invagination of the neuroectoderm to develop the neural groove and the neural tube.
In the anterior region of the neuroectoderm, molecular signals designate a subset of cells known as the eye field.
Here is the development of the neural tube, and this is the anterior portion of the area of the forebrain that will later on form the optic vesicle. So both of optic vesicles on both sides will laterally grow until they reach the surface ectoderm.
Originating as a single group of cells, lateral migrations separate the eye field into 2 groups, or eye primordia. This lateral migration can be seen at day 22 of human embryonic development.
At the 3rd week.
Small indentations termed the optic grooves. As these cells migrate further into the surrounding mesenchymal tissue, they form balloon-like outpocketings known as optic vesicles. Contact with the surface ectoderm initiates inductive events between these tissues, causing the optic vesicles and overlying ectoderm to invaginate, or migrate back toward the underlying mesenchyme.
So there will be invagination of the optic vesicle upon the contact with the surface ectoderm.
This process results in the optic vesicle forming a double-layer tissue known as the optic cup.
So the optic vesicle will be transformed into an optic cup, and thus this optic cup will share in the formation of different ocular structures that is mainly formed of an outer layer and the inner layer.
The outermost layer becomes retinal pigment epithelium and optic stalk, while the innermost layer...
So as you can see here, this is the optic cup is formed, and forming the outer layer, forming the RPE, and while it is fused together to form the stalk, or what's called optic stalk, that is connecting the optic cup to the forebrain, it will develop later on the optic nerve.
Becomes the neural retina. Formation of the lens, iris, cornea, choroid, and sclera.
So here are the development of the ocular primordia from the neural crest cells and from the neural tube as an outpouching of the neural tube, and until they reach the surface ectoderm to induce some changes like development of the lens from the surface ectoderm and development of optic cup and invagination of the optic cup that will later on form the iris and ciliary body and the retina, and while the part of the part that connected the optic cup to the forebrain will form later on the optic nerve, what's called the optic stalk. So here we have the we will now study the more specifically the development of each ocular structure. So we'll start first by the anterior segment development, and in the second half we will start or study the development of posterior segment and the blood vessels. So the anterior segment development, here we have the section of the anterior segment. All of these anterior segments are initiated by the contact between the optic vesicle and lens placode. So as the ocular primordia is developing, it will touch the surface ectoderm, as we mentioned, leading to a series of changes within the surface ectoderm forming the lens, and a series of changes within the ocular primordia to form different structures of the anterior segment. So here what we will discuss in the first part, which will should be six structures forming the anterior segment. It will be we study first the lens and the cornea, anterior chamber and iris, ciliary body, and finally the posterior chamber. So are you ready? Do you have any question in the previous part? Do you have any question in the introduction? No answer? Hello? Can you hear me? Okay. So let's start immediately with the anatomy of the or the embryology of the anterior segment structures. Let's start first with the lens. So as we mentioned, the lens will be derived from the surface ectoderm. So as you can see here, this is the developing optic vesicle, and as it grows laterally, it will touch the surface ectoderm, and this touch will initiate the development of the lens, which is called the lens placode or lens plate. This occurs in about the 27th day of gestation. So this is later on will lead to this touch will lead to invagination of the optic vesicle, leading to formation of what's called the optic cup, which has an inner layer of the optic cup and outer layer of the optic cup. Since the edges of the optic vesicle has transformed into the optic cup, this will lead to formation of a cavity inside the optic cup. This cavity will be occupied by the developing lens. So the lens will, or the lens placode will invaginate inside the cavity of the optic cup, leading to formation of what is called the lens vesicle. Later on, there will be contact between the edges of the optic vesicle, leading to separation, leading to formation of what's called the lens vesicle. So this occurs in about the 33rd days. And if you think this will be logical, so if we have here, this is the developing eye, and this is the developing lens, and as you can see here, the developing lens is outside the eye. So how could this be possible? This will be because that this is surface ectoderm will invaginate inside the developing optic cup, leading to presence of the lens vesicle within the optic cup. So the lens has become now an intraocular structure. So again, here's a section of the developing optic disc. So the developing optic vesicle. So they will be transformed into an optic cup. It has a central pit that's called optic pit. Once it is in contact with the surface ectoderm, this will lead to transforming this lens ectoderm into lens pit and lens placode that will initiate a series of changes within the optic cup, leading to formation of invagination of the optic cup and invagination of the lens vesicle inside, and then separation from the surface ectoderm to form the primary lens vesicle. So the lens vesicle, if we have a closer look at the structure of the lens vesicle, we will find that the lens has anterior capsule, anterior epithelium, which is a cuboidal epithelium, and it has it should have the posterior epithelium. However, the posterior epithelium, that was a columnar epithelium, will be consumed, as you can see, by development of the primary lens fibers. So the primary lens fibers are made by by elongation of the posterior lens epithelium that will elongate to occupy the space or the interior of the lens cavity. The lens epithelium will secrete type 4 collagen and the glycosaminoglycans to form the lens capsule. And this, the primary lens fibers or the anterior lens epithelium will later on form the secondary lens fibers. So you remember this from the anatomy of the lens. As we mentioned, we said that as we grow old, we have the development of lens fibers, and development of lens fibers, these are secondary lens fibers that will develop from the equatorial lens that is coming from the anterior lens epithelium. However, as an adult or even immediately after birth, we don't have posterior lens epithelium because it was consumed in the embryonic life by development of the embryonic lens, as you can see here. So again, the cells of the posterior lens epithelium will elongate to occupy the space inside the lens, and they will secrete lens protein, which is the crystalline that is responsible for lens transparency. So this occurs, as you can see here, in the first 2 months. The cells of the posterior epithelium will continue to grow until they fill the whole cavity of the lens, and this is now called the embryonic nucleus. So the embryonic nucleus is the nucleus formed in the first 2 months of gestation by the elongation of the posterior epithelium of the lens. So here again, we can see that the secondary the fibers or the primary fibers are formed by the posterior epithelium. So there is no posterior epithelium here, while in the secondary fibers are formed later on by the primary lens epithelium at the equator. So this is can called now the fetal nucleus. So the fetal nucleus is formed by after the second month from the secondary lens fibers which lay down from the anterior lens epithelium. So please concentrate well and know what is the source of every fiber. So the embryonic nucleus is derived from the primary lens fibers that are derived from the posterior lens epithelium, while the fetal nucleus is derived from the anterior lens epithelium that will form the secondary lens fibers. The junction between the anterior and the posterior edge of the fibers will form what is called the Y-shaped suture. So we have an erect Y suture that's present anteriorly, and we have an inverted Y suture which is present posteriorly. So basically, the lens at birth, it's about 90 milligram in weight, while it continues to grow in size as it reaches in the adult size will be 255 milligram with the thickness of about 3.5 milligram. So the lens fibers is formed of different nuclei that are developing throughout the life, starting from the early gestation. So we have first the embryonic nucleus derived from the primary lens fibers in the first 2 months. Then it will around the embryonic nucleus, it will be the fetal nucleus, which will continue to grow until the delivery or until birth. Then from the birth till the adulthood, till about 25 years old, there will be the infantile nucleus, which is this is the blue one, and from the adulthood, for all life, for the entire life, there will be development of the adult nucleus, as you can see here. So the lens fibers forming process is a lifelong process that occurs from the first month of gestation for the entire life. We need to know some basic information about the congenital anomalies related to the developmental defect in the embryogenesis. So we don't need to know all this information, just to know where or as in the basic level or in the basic exam, you need just to know where is the defect or what is the basic or what is the embryological basis of the congenital anomaly. So you don't need to know what are the manifestations or what are the clinical features or what is the inheritance. So you just need to know what is the reason behind this congenital anomaly. So sometimes we have a congenital anomaly in the lens, like aphakia. So the child is born without a lens. This can be either primary or secondary, and the secondary is much more common than the primary. So why does this occur? This occurs because of failure of touch between the surface ectoderm and the optic vesicle. So this touch will induce the formation of lens. If there is no touch between the surface ectoderm and the optic vesicle, there will be no lens. However, as I told you, there will be a secondary aphakia. It's more common, and this secondary aphakia occurs due to spontaneous resorption of the already developed lens. Sometimes there will be abnormalities or congenital anomaly regarding the curvature of the lens. So they may assume a conical shape like lenticonus or take a spherical shape like a lentiglobus. So these can occur with a famous syndrome. It's called Alport syndrome. But in case of Alport syndrome, there will be associated with anterior lenticonus rather than posterior lenticonus. An important congenital defect, or the most important congenital defect in the perspective of the basic exam, will be the lens coloboma. So the coloboma meaning that there will be a defect. This defect in either in any tissue, such as the lens or the iris or the choroid or the optic nerve or the retina. This all occur due to one reason: failure of closure of embryonic fissure, as we will see at the end of lecture. So lens coloboma occur present with a defect in the lens, with defect in the zonule, and can be associated, can be either isolated or associated with other congenital colobomas. And it is most probably or most commonly present inferior nasal, and it is due to failure of closure of embryonic fissure. The most common congenital anomaly of the lens will be the congenital cataract. So congenital cataracts are mostly idiopathic but can occur due to a wide range of reasons like infections or metabolic abnormalities. So this is beyond the scope of a basic exam, but this is just to know that this is the most common congenital anomaly. Okay. Let's start now with the second structure of the anterior segment, which is the cornea. So the development of the cornea coming from the surface ectoderm. And again, this is logic because we have, this is the lens, and this is now the lens is separated from the surface ectoderm, and the structure that is anterior to the lens should be the cornea. So the cornea is derived from the surface ectoderm in front of the developing lens vesicle. And this surface ectoderm will mainly, after separation, this will mainly form the epithelium of the cornea. So the epithelium of the cornea is derived from the surface ectoderm. So please notice that every layer within the cornea is derived from a different structure. So we are starting now by the lens epithelium. So the lens epithelium will be derived from the surface ectoderm. Then the basal lamina of the surface ectoderm or the basal lamina of the epithelial cells will secrete some collagen fibers and glycosaminoglycans, which will form the primary stroma. So as we know, the primary stroma is formed from three structures. It will be formed from collagen fibers, glycosaminoglycan matrix, and the keratocytes. The collagen fibers and the glycosaminoglycan matrix is derived from the epithelium of the cornea. So it will form the primary stroma. So this is not will be the definitive or not the final stroma. So corneal embryogenesis will continue on the 5th week by some what's called mesenchymal waves. So there will be waves of mesenchymal cells or the neural crest cells and the mesoderm that will share in the formation of the anterior segment of the eye. So there are three waves that will migrate toward the developing eye. The first wave, or the first mesenchymal wave, will be situated behind the developing corneal epithelium and the primary stroma, so they will form the corneal endothelium. So it will be situated between the lens and the corneal epithelium, as you can see here. So the first wave will form the corneal endothelium. And later on, this endothelium will secrete the Descemet membrane. So the Descemet membrane will be secreted or will be the basal lamina of the endothelium. So this will be the first wave of mesenchymal cells. Then the third wave of the mesenchymal cells will be between the epithelium and the endothelium, and this what will form the keratocytes. So remember that we mentioned that the epithelium will secrete the primary stroma that is devoid from the keratocytes. So what will be the source of keratocytes? Yes, it will be coming from the third mesenchymal wave. So the third mesenchymal wave will occupy the area of stroma, sharing in the formation of stroma by supplying the keratocytes, and here we have the final stroma. So here we have a question. Oh, you said just the first mesenchymal and then the third mesenchymal. So is it a mistake? It should be the second mesenchymal wave. The answer is no. The second mesenchymal wave will share in the formation of another structure that we will say later on. So the keratocytes will secrete type 1 collagen fibers and proteoglycan, which will form the lamellae, which are organized into lamellae that will form the stroma of the cornea. So to summarize, the corneal diameter at birth will be 9.5 to 10.5 millimeters that will reach the adult size at age of 2 years. And to summarize the different derivatives of the layers, so we have first the epithelium that is derived from the surface ectoderm, and the basal lamina of the epithelium will secrete the primary stroma, which will be developed, which will be formed from the collagen fibers and the glycosaminoglycan. So they are developed from the surface epithelium. Then the primary mesenchymal wave, or the first mesenchymal wave, which will be formed from the neural crystals, will form the endothelium, and the endothelium, the basal lamina, will secrete the desmet, and then the second mesenchymal wave will form the keratocytes that will secrete the collagen type 1, and that will form eventually the definitive configuration of the corneal lamellae and form the definitive stroma. So this is hot topics that will be asked in the MCQ exams. So to know that the endothelium is derived from the first mesenchymal wave, while the third mesenchymal wave will form the keratocytes. So speaking about the size of the cornea, or the normal size of the cornea, we need to know what are the abnormal sizes of the cornea. So sometimes the cornea could be small, meaning that it is less than 9 millimeters in the newborn or less than 10 millimeters in the adult. Or sometimes it can be large, or what's called megalocornea, which meaning that the cornea is more than 12 millimeter at birth or more than 30 millimeter in adults. So why do they occur? This is like it is due to disproportionate space between the developing optic tips of the optic cup and the space between the optic cup and the lens, meaning that if there is failure of the corneal growth in the 5th month occurs due to overgrowth of the anterior tip of the optic cup, so there will be no space for the cornea to develop. On the other hand, the megalocornea, meaning that there will be failure of the optic cup to grow, leaving large space to the cornea to develop. And let me show you this in an illustration. So here is the optic cup. And here is the developing lens, or the optic vesicle. If the optic cup, or if the optic cup edges are too large, so they are becoming like this and like this, there will be too limited space like here for developing of the cornea, so the cornea will be very small. And on the other hand, if the edges of the optic cup are small like this and this, so the area for developing of the lens, or the developing of the cornea, will be very large, because the cornea will develop between the both edges of the optic cup. So congenital anomalies regarding the size of the optic cup of the cornea is mainly related to the space between the optic cup between the two edges of the optic cup. Overgrowth will lead to smaller cornea, while the rest of the growth will lead to larger cornea. Okay, so the sclerocornea can occur due to present scleronization of the cornea, and the cornea become opaque and will ill-defined limbus. So you will not be able to distinguish between the cornea and sclera. And this is occurred due to failure of the second mesenchymal wave migration. We will see about it later, and we will know what will the second mesenchymal wave will form in the eye. Sometimes there are other dystrophies like corneal dystrophy. So the corneal dystrophy or endothelial dystrophy present with a ground glass appearance of the cornea with clear periphery, and this is occurred due to abnormal endothelium. So since the endothelium is formed from the primary mesenchymal wave, so this will be the site of lesion. There will be a dysfunction of the primary mesenchymal wave. Keratoconus can be assumed by many authorities as a hereditary disease. So it has a genetical basis because it can run in families. So in this disease, the cornea assume a conical shape, and there will be bulging of the corneal stroma and the fragmentation of Bowman's membrane, and sometimes this can lead to breaks in the Descemet membrane, leading to gush of aqueous inside the cornea, causing acute hydrops. So this can be also considered one of the congenital anomalies of the cornea. Okay, so now we will come to the development of the anterior chamber and the angle. So we have just mentioned, or we discussed the development of the cornea and the lens. Let's now discuss the development of the anterior chamber and the angle. So this occurs, or the development of the anterior chamber starts by the third week by successive by the three mesenchymal waves that we just mentioned. So the three mesenchymal waves are surrounding the optic cup, and they will be responsible for formation of different structures. For example, we said that the first mesenchymal wave will form the corneal endothelium, the third wave will form the keratocytes of the cornea, and here we come to the second wave, which will form the pupillary membrane. So the development of the anterior chamber will be initiated by the three mesenchymal waves. So how could this be possible? This will be by development of the what are the boundaries of the anterior chamber and the angle. So it is bounded anteriorly by the endothelium and posteriorly by the iris, which will be fused, forming a pupillary membrane posteriorly. So the anterior chamber will be formed by a slit-like space between the corneal endothelium and the iris epithelium as a result of mesenchymal atrophy. So here we can see this is the developing lens. As you can see, these are the posterior lens fibers or the primary lens fibers, and this is the developing edges of the optic cup, formed of the inner layer and the outer layer. This is the outer layer, this is the inner layer, and this is the outer layer, and this is the developing cornea. So here is the endothelium, and we have three mesenchymal waves occupying surrounding the area of the optic cup. So these mesenchymal waves will undergo atrophy in the area between the endothelium and the iris, which is formed from the distal part of the edges of the optic cup. So this will lead to developing of the anterior chamber. So again, the first mesenchymal wave will form the endothelium, and the second mesenchymal wave will form the pupillary membrane, which will be closing the pupillary opening in the early embryonic life until the 8th month, while the third wave will form the keratocytes. So what happened here is that there will be a slit-like atrophy or cleavage within the mesenchymal cells in between the corneal endothelium and the pupillary membrane. So what will be the sequence of events? At the 15th week of gestation, the corneal endothelium will extend here into the angle and forming the angle recess to meet the iris. So the iris and the corneal endothelium will meet together by the 15th week of gestation. And by the third month, the angle recess more deepen and form the iridocorneal angle. At the 7th month of gestation, there will be a structure here that is called the trabecular meshwork. So this is mainly, it is a mesenchymal cells again. These are the mesenchymal neural crest cells that will develop later on the trabecular meshwork, as we will see later. After this, the canal of Schlemm will develop from a small plexus of venous canaliculi, which has an endodermal origin. So please note that mostly the blood vessels are formed from the endoderm. So the canal of Schlemm will be developed from the endoderm. So as the trabecular meshwork is continue to recede 6 to 12 months after gestation, it becomes the adult type appearance. So the anterior chamber will be about 2.5 in diameter, while in the adult it's 3 millimeter in diameter. In the final weeks of gestation, the trabecular meshwork here show the fenestration. So during the embryonic life, the trabecular meshwork don't show the fenestrations that will communicate between the anterior chamber and the canal of Schlemm. These fenestrations only develop within the final week of gestation. Sometimes failure of formation of this fenestration within the trabecular meshwork will lead to formation of congenital glaucoma. So congenital glaucoma meaning that there is dysgenesis of the trabecular meshwork, and there is the trabecular meshwork openings haven't developed properly, leading to trapment of the aqueous in the anterior chamber, causing rise of the intraocular pressure. Okay, so let's now talk about the development of the iris and ciliary body. At the end of the third week of gestation, there will be two layers of the neuroectodermis. So the layers of the optic cup are called in the neuroectoderm. It's formed of the outer layer and inner layer. This is the lids, and this is the edges of the optic cup. It's called the neuroectoderm. So the outer layer will form the pigmented epithelium, while the inner layer will form the non-pigmented epithelium. The distal part here will form the iris. So the distal part of the edge of neuroectoderm of the optic cup will form the iris, while the proximal edge will form the ciliary body. So here is the iris, and here is the ciliary body. So they are formed of pigmented and non-pigmented layers. Both of them will form the neuroectoderm of edge of the optic cup. So there will be cellular proliferation of the proximal two layers of the neuroectoderm. So here, the proximal two layers of the neuroectoderm will show some longitudinal indentation of the outer pigmented epithelium. So the outer pigmented epithelium proximally at the area of the ciliary body will show indentation. And this underneath this indentation, at the age of 12 weeks, there will be forming radial fold beneath this indentation these radial folds will form the ciliary process. So the ciliary process are formed as radial folds at the 12 weeks of gestation. At 10th week, or at week number 10, the mesenchyme around the area of ciliary body here will form the stroma of the ciliary body. So the mesenchymal cells will form the stroma of the ciliary body, while the ciliary muscle will develop from at the 12th week. The myofilaments for proliferation of mesenchyme will form the smooth muscles of the ciliary body. And the ciliary muscle will continue to develop at least 1 year after birth. That's why it will be difficult to dilate the newborn, as his muscles are underdeveloped. So here is again at the region of the ciliary body, there will be development of the it will be start with proliferation of the neuroectoderm forming the outer pigmented epithelium. So the outer pigmented part of the ciliary body will be formed from the neuroectoderm. Then the inner layer will be formed, or the inner non-pigmented layer will be formed, or will form the ciliary process. Then at the area of the ciliary body, the stroma will be formed from the mesenchymal cells, and again the mesenchymal cells will share in the formation of the ciliary muscle, which is present at the ciliary body. So this is about the anatomy. What about the function? So the ciliary body function will be mainly to secrete the aqueous humor. So secretion of the aqueous humor or pumping of the aqueous humor starts by the 4th month of gestation. So by 4th month of gestation, the aqueous will be filling the anterior chamber and posterior chamber, while the ciliary epithelium will start to synthesize collagen fiber, which will become the suspensory and the suspensory ligaments or zonule of lens, in addition to the tertiary vitreous. So both of them will share in the formation of the suspensory ligaments of the lens or zonule of the lens. Speaking about the development of the iris starts in developing at the 3rd month. As we mentioned, the distal edge of the developing optic cup or the neuroectoderm will form the outer layer and the inner layer. So it will form the pigmented layer and the non-pigmented layer. The pigmented layer will share in the formation of the iris pigment epithelium, while the mesenchyme will form the anterior stroma. So the stroma of the iris will be formed by the mesenchyme, and the neuroectoderm of the optic cup will be forming the iris pigment epithelium and the sphincter and dilator pupillae muscle. So this is one of the common questions to confuse you in the exam. So you think that all muscles are developing from the mesoderm, like extraocular muscles, for example, but then sphincter and dilator pupillae muscles are developing from the neuroectoderm. So when does this iris start to form? This occurs by the initiation of the second wave. So we said that the second wave of mesenchyme will form the pupillary membrane. Once the pupillary membrane is formed, it will be fusing with the iris, leading to development of the iris. So let's see. So the pupillary membrane will start as condensation of the second wave of mesenchymal cells. As you can see here, this is the edge of the optic cup, and here is the developing lens, and this is the mesenchymal cells in between and around the developing eye. So here is the optic cup, and here is the developing cornea, and here is the developing lens. So the area in between will be the condensation of mesenchymal cells that will form the 3 waves, as we mentioned. So the second wave will form the pupillary membrane, in extending in front of the behind the cornea and in front of the edges of the optic cup and the optic vesicle. As you can see here, this is a section of eye of a developing embryo, showing the developing optic vesicle and the optic cup, and here is the pupillary membrane. So during embryonic life, we didn't have a pupillary aperture or pupillary opening like as an adult, but this will develop later on in the gestational life, it will develop in the 8th month, not in the 3rd month. So the pupillary membrane starts in the 3rd month, and the 2 layers of the neuroectoderm from the optic cup will extend posterior to the pupillary membrane. So if we have here, this is the pupillary membrane. Once it is formed, the distal part of the neuroectoderm will be extending behind, as you can see here, extending behind the neuroectoderm to form the developing iris. And this structure will fuse, so the pigmented layer and the non-pigmented layer and the pupillary membrane, all these structures after extension will form the iris. So the iris will be formed of the pupillary membrane from anterior to posterior pupillary membrane, then non-pigmented epithelium, and then pigmented epithelium, then non-pigmented epithelium. So we have now a closed iris. So what will happen later? So at the 3rd month of gestation, the anterior epithelium or the pigmented epithelium of the neuroectoderm will differentiate to form the sphincter and the dilator pupillae muscle. As you can see here, this is the anterior epithelium of the neuroectoderm will share in the formation of the dilator and the constrictor pupillae muscle. The pupillary membrane itself will differentiate and form like collagen fibers and extracellular matrix, which will form the anterior stroma. So the anterior stroma of the iris will be formed from the pupillary membrane. The pigmentation of the posterior epithelial layer occur at the pupillary margin at the midterm. By the 7th month of gestation, the iris is fully pigmented. So pigmentation of the iris start at the midterm, and about 7th month of gestation, the iris will become fully pigmented. Here we can see this is the iris stroma formed by the pupillary membrane. And here is the pigmentation or the posterior the posterior epithelial layer will or posterior epithelial cells will be pigmented at the pupillary margin at the midterm. Then at the 8th month of gestation, the pupillary membrane starts to degenerate. So we have here, this is the iris, and this is the persistent pupillary membrane. At the 8th month of gestation, this will disappear. So there will be an opening within the center of the iris. So this opening will be now termed the pupil, and the iris stroma and dilator pupillae muscle will be still immature at birth. That's why usually the infant or the newborn will be meiotic, because of the immature muscles, and the sphincter muscle will be more strong, taking the upper hand, so that the pupil mostly will be meiotic, and it is more difficult to dilate the newborn than the adults. Now we come to some congenital anomalies of the iris. So we have sometimes the iris that will not develop, causing a hypoplasia or aplasia. And this is again like due to inadequate interaction between the optic cup and the surface ectoderm and the neural crest cells. And usually this occurs due to defect in a famous group of genes called the PAX6 genes. So if there is no space or no interaction between the optic cup and the neural crest cells, that will form the secondary wave of mesenchyme and the surface ectoderm, so there will be lack of development of the iris. Another congenital anomaly we said is the persistent pupillary membrane. So as we mentioned, the pupillary membrane should degenerate at the 8th month of gestation. If it didn't regenerate, then this will lead to persistence of this membrane as a congenital anomaly. Usually it doesn't have to be that large, and sometimes there are only very small remnants from it that are not visually significant. Again, we come to the coloboma. So coloboma occurs due to failure of the embryonic fissure to close, and the appearance of the teardrop appearance of the iris, which is most probably due to defect in the iris tissue, more commonly at the infranasal quadrant, that can be associated with other colobomas of the eye of different ocular structures, and they can also be associated with systemic diseases like Charge syndrome. So Charge syndrome meaning that there is ocular coloboma, heart defect, choanal atresia, mental retardation, genitourinary and ear anomalies. This is called Charge syndrome. Other congenital anomalies of the iris like polychoria, which meaning that there are accessory iris opening, which occurs due to anterior segment dysgenesis, like Axenfeld-Rieger syndrome. Correctopia, meaning that there is displacement of the pupil, which can occur due to hypoplasia or due to lens subluxation, such as in case of ectopia lentis e pupillae. A small pupil or microchoria or congenital meiosis can also occur due to maldevelopment of the dilator pupillae muscle, and on the reverse, the congenital mydriasis can occur due to maldevelopment of this iris sphincter muscle. The last part of the anterior segment will be the posterior chamber. So how does the posterior chamber develops? So the posterior chamber will develop as a slit again in the mesenchyme, but not in this time not anterior to the developing iris, but it will be posterior to the developing iris and anterior to the developing lens. So the area occupied by posterior chamber will have mesenchymal cells. The mesenchymal cells will undergo selective atrophy at this area, leading to formation of the posterior chamber. Then when the pupillary membrane disappear, there will be communication between the anterior and posterior chamber, and after secretion of the aqueous, the aqueous will circulate freely between posterior and anterior chamber through the pupil. So here we finished the development of the anterior segment. Do you have any question in the development of the anterior segment? Okay, I guess that there are no questions. So is everything clear? Please tell me if anything is not clear. I know that embryology is not an easy topic, so please let me know if you have any doubts or if you have any topic or any area that is vague to you. Okay, I guess that there are no questions. So, okay, we will carry on the second part, which will be the development of the posterior segment. So, the development of the posterior segment and the posterior part of the eye will be including the retina or the optic nerve, the vitreous, the choroid, the sclera, and finally we will finish by the vascular system. So, starting by the retina, how does the retina develop? So the retina is formed, as we know, from an outer layer is called the retinal pigment epithelium, and the inner 9 layers they are called collectively the neurosensory retina. So as we saw in the video of the development of the eye, the inner 9 layers of the retina or the neurosensory retina is developing from the inner layer of the optic cup, or inner layer of the neuroectoderm of the optic cup, while the retinal pigment epithelium will be developing from the outer layer or outer neuroectoderm from the developing optic cup. So let's start to know what is the differentiation of the neurosensory retina and how does they develop each type of cells. Please concentrate with me because sometimes this can be a bit confusing. So here we have a section of the developing eye. Here are the edges of the optic cup, and here is the developing lens. So this is the inner layer of the optic cup that will form the neurosensory retina, and this is the outer layer which will form the retinal pigment epithelium. As we mentioned in the anatomy or the embryology of the anterior segment, we said that the distal part of the edges of the optic cup will form the iris and ciliary body. So the distal part will form the iris, and here is the proximal part will form the ciliary body. So the retina will be developed from the posterior 4/5. The posterior 4/5 of the inner layer or the inner layer of the optic cup. This what will form the primordial sensory retina. So again, the sensory retina will be formed from the inner layer of the posterior 4/5 of the optic cup. By the 7th week, there will be differentiation inside the inner layer of the optic cup into 2 layers. The inner layer or inner one-third, it is called the marginal zone because it is near the margin. So this is called the marginal zone. And this marginal zone will have no nuclei, and this marginal zone will form later on the nerve fiber layer of the retina. So the inner one-third of the marginal zone of the inner layer of the optic cup will form the marginal zone. However, the outer two-thirds of the inner layer of the optic cup will form the primitive nuclear zone. So this primitive nuclear zone will form later on the nucleated cells that will form the neural cells of the retina. So here is a favorite question in the exam to ask you that where does the primitive nuclear zone develop from? Is it from the inner layer of the inner optic disc, or from the inner zone of the inner layer of the optic cup, or from the outer zone of the inner layer of the optic cup? So this comes from the outer zone of the inner layer of the optic cup. So you just got confused in the exam, and you think that, okay, I know that the retina is coming from the inner layer of the optic cup and the RPE is coming from the outer layer of the optic cup. So sure that the nuclei are coming from the inner zone of the inner layer of the optic cup. So this is wrong. The inner layer or the inner zone of the inner layer of the optic cup will form the nerve fiber layer. However, the outer zone of the inner layer of the optic cup will form the nuclear cell layer. I hope that this concept is clear to you. Going further into the neurosensor retina, so we said that it developed from the primitive nuclear zone. So here is the developing eye, and here is the primitive nuclear zone from the outer zone of the inner layer of the optic cup. This primitive nuclear zone will undergo differentiation to form the nuclear and the glial cells that will be later on distinctive into two zones. The outer zone will be the outer neuroplastic layer forming the photoreceptor. So again, the outer neuroplastic layer, which will be the outer zone of the outer primitive nuclear zone, will form the retinal pigment epithelium. So we remember that it is outer. So the photoreceptor, it is outer because it is in close relation to the choroid. So it will be outer. While the inner neuroplastic layer will form the ganglion cell layer. And the ganglion cell layer is more inner toward the nerve fiber layer. So it is developed from the inner neuroplastic layer. Separating both the outer zone and the inner neuroplastic layer, there will be a transient nerve fiber layer that's called the transient nerve fiber layer of Chevitz. This transient nerve fiber layer of Chevitz will later on will form the inner plexiform layer. So again, the outer nuclear zone or the primitive nuclear zone occupying the outer zone of the inner layer of the optic cup will later on differentiate into inner zone and outer zone. The outer zone will form the photoreceptors, while the inner neuroplastic layer will form the ganglion cell layer, separating both of them a transient layer that's called the transient nerve fiber layer of Chevitz that will later on differentiate into the inner plexiform layer. Then this will be followed by further differentiation of the neuroplastic layer to form the photoreceptors like rods and cones, and differentiation of the outer nuclear layer to form bipolar, amacrine, and the horizontal cells, and to form the inner nucleated layers of the retina. The ganglion cell, which will be derived from the inner neuroplastic layer, will form the ganglion cell layer, and the axons of this ganglion cell will form the nerve fiber layer, or the primitive nerve fiber layer. Then the retina will show different cells and synapse formation. So here is the developing optic vesicle. Here is the optic cup, and this is the inner marginal zone form the nerve fiber layer, and this is the outer marginal zone or the primitive nuclear zone, which is located outer, that is has an inner neuroplastic layer forming the ganglion cell layer and the outer neuroplastic layer forming the photoreceptors, and further differentiate into other nucleated cells like bipolar, amacrine, horizontal cells, and but separated between both of them by the transient layer of Chevels, which will form the inner plexiform layer. So this will be the configuration of the primary neurosensory retina. Serial proliferation and the melanogenesis, so there will be melanin deposition within the outer wall of the optic cup, will occur at the 6th week to form the retinal pigment epithelium. And by the 15th week, all cell types like all cells and synapses are formed within the neurosensory retina, while the fovea is formed by the thinning of the ganglion cell layer and the inner nucleated layer by 24th week of gestation. And by 5th week, this will be the configuration of the retina. The retinal pigment epithelium will be derived from the outer layer of the optic cup, while the neurosensory retina will be derived from the inner layer of the optic cup, formed by the outer segment and the photoreceptors, outer nucleated layer of the retina, then outer plexiform layer, then inner nucleated layer, which is formed by the horizontal, bipolar, and the amacrine cells, then the inner plexiform layer, then the ganglion cell layer, and finally the nerve fiber layer. So remember that the outer nucleated layer are derived, all these are derived from the outer neuroplastic layers. So all nucleated layers except the ganglion cell layers are derived from the outer neuroplastic layer, while the ganglion cell layer will be derived from the inner neuroplastic layer. Let me just this. Okay, so this is the development of the retina. Let's now move to the development of the optic nerve. So the optic nerve developed from the optic stalk. So, and the optic stalk will be the connection between the optic vesicle and the forebrain, as we mentioned before. So this is the developing optic vesicle, and this is the forebrain, and this will be the elongation of the optic stalk is what will remain the optic nerve. If we had a cross section here within the optic nerve, within the optic stalk, then it will has two layers, inner layer and outer layer. Here will be a section in the developing optic stalk. So it will have inner neuroectodermal layer here and outer neural crest layer there. So here is the inner neuroectodermal layer, and here is an outer neural crest cell layer. So in the 6th week, the inner layer of the nerve fiber, so here is the inner layer of the optic stalk will degenerate. So here will undergo degeneration and become vacuolated. This is to allow space for the nerve fibers or the axons from the ganglion cells to enter through this vacuolated space and form the opening and form, or to allow some opening for the nerve fiber to enter. Then at the 33 weeks of gestation, the optic nerve assume the adult size and the adult configuration of the optic nerve, consisting of about 1.1 or to 1.2 millions of nerve fibers or millions of axons of nerve fibers. At the 8th week of gestation, there will be the inner layer. A few cells of the inner layer here will differentiate into glial cells, and these glial cells will form the lamina cribrosa. This is all coming from the inner layer. So the inner layer of the optic nerve will form all these structures. They will undergo degeneration to allow passages of the nerve fiber layer and will form the glial cells that will form the lamina cribrosa. So what about the outer layer? The outer layer of the optic nerve will form the glia. So the glia, will form the meninges. So it will form the pia, the arachnoid, and the dura mater, which will surround the optic nerve. So the outer layer will share in the formation of the sheath of the optic nerve. Well, this occurs by the 4th month. So here is again, these are the ganglion cells, and these are the nerve fiber layer, and this is the developing optic nerve. So the optic nerve is developed from outer layer, which is formed from the neural crystals, and inner layer, which is formed from the neuroectoderm. The inner layer will undergo vacuolation to allow passages of the nerve fibers from the developing ganglions, from the developing retina, and also will form the glial cells that will form the lamina cribrosa. However, the outer layer, which will be formed from the neural crystals, will develop the optic nerve sheath. Now, speaking about the congenital anomalies of the optic nerve, so we have the morning glory syndrome. So the morning glory syndrome, the disc appears large and excavated, funnel-shaped, surrounded by a halo, and it has some blackish pigments, and there will be radiating blood vessels from the optic disc as if it is like a sun. So this occurs due to abnormal closure of the embryonic fissure. Coloboma of the optic nerve can also occur due to a failure of closure of the embryonic fissure. It can be either isolated anomaly or associated with other colobomas of the eye, as we just mentioned, and it can be also associated with systemic disease such as Charge syndrome. Now let's talk about the development of the vitreous. And development of the vitreous, it is one of the most important topics in the embryology. So if you told me, or if you asked me, what are the most important topics in the embryology, I will tell you it will be the development of the lens and the vitreous and the cornea. So these are the most important topics within the embryology. And most of the questions are circulating between these topics. So back to the development of the vitreous. So the vitreous will develop at the space between the optic cup and the lens. They are derived from the mesoderm with some contribution from the ectoderm. The development of the vitreous takes this importance because it develops into 3 stages. So you need to know what are the stages of vitreous development and what will be the derivative of each stage. So let's start first by the primary vitreous. So the primary vitreous starts at the 1st month of gestation. There will be some fine, delicate cytoplasmic process that occupy the space between the lens and the inner layer of the optic cup. So these delicate cytoplasmic process will form the primary vitreous. It is composed mainly from the ectoderm, the fibrils coming from the ectoderm, and the mesenchymal cells or mesoderm, which will form the primary vitreous. So the primary vitreous is formed from the ectoderm and mesoderm. And here you can see there is a blood vessel. This blood vessel, it is called the hyaloid vessels. These hyaloid vessels will be supplying the primary vitreous, and it will enclose the developing lens vesicle. So the primary vitreous will be present in, or the hyaloid vessels are present in the primary vitreous. Later on, there will be the development of the secondary vitreous. So the primary vitreous start to atrophy, or in the 2nd month, you will see there will be a fragmentation of the hyaloid vessels, and the primary vitreous will differentiate to the next level, which will differentiate into the cells called hyalocytes. These hyalocytes are the primary cells of the vitreous that will secrete type 2 collagen and secrete hyaluronic acid, which will form the secondary vitreous. So the source of secondary vitreous will be from the hyalucytes. And the secondary vitreous is the adult vitreous. Here is the trick. The secondary vitreous is the vitreous like a vascular gel substance that occupy the space between the lens and the retina. And further, at the 5th to 6th months of gestation, there will be a complete regression and atrophy of the hyaloid vessels here. And sometimes they will leave a canal at the center that's called the canal of Cloquet. So again, the secondary vitreous is derived from hyalucytes that will secrete type 2 collagen, which is the main collagen fibrils of the vitreous, and will secrete the hyaluronic acid, which will form the avascular gel-like structure of the vitreous. So the secondary vitreous is the permanent vitreous or the adult vitreous. So you may wonder, so what will be the tertiary vitreous stands for? If the secondary vitreous will be the final vitreous or the adult vitreous, what will be the rule of the tertiary vitreous? Well, the tertiary vitreous is just a condensation at the area of the zonule between the ciliary body and the lens capsule. So the zonular fibers are made by the tertiary vitreous. So again, the tertiary vitreous will form the zonule or the lens zonule. So collagen fibrils synthesized by the ciliary epithelium will become more condensed here and extend to the lens equator, and that will make the tertiary vitreous. So to say it for the final time, or this is the tertiary vitreous will be making the lens zonule, not participating into the vitreous body itself. Congenital anomalies of the vitreous, we have a famous congenital anomaly of the vitreous that is called the persistent hyperplastic primary vitreous. So the primary vitreous, it should absorb and it should atrophy and be replaced by the secondary vitreous. In some occasions, there will be failure of this primary vitreous to atrophy or to regress. So the primary vitreous has the hyaloid vessels. So both the primary vitreous and the hyaloid vessels are failed to regress, and this is usually non-hereditary, so it is not associated with systemic defects, but associated with other ocular defects like small eye or microphthalmos and cataract and leukocoria. So these patients will develop with leukocoria. Okay, so now let's talk about the development of the choroid. So the choroid is a vascular endothelium, and the hematopoietic cells of the choroid are derived from the endoderm. So as I told you when I was talking about the canal of Schlemm, I told you that mostly the blood vessels are derived from the endoderm. So the blood vessels are derived from the endoderm, while the choroidal stroma formed from the pericytes, smooth muscles, the melanocytes, and the collagen are derived from the ectoderm. So here is the choroid. Here is the edges of the optic cup, and the choroid will be formed by condensation from neural crystals around the optic cup. So again, the choroid will be developed from the neural crystals or mesenchyme. Differentiation of the neural crystals will form the choroidal stroma by the end of the 3rd month of gestation. So here is the choroid, and the differentiation of the neural crystals here at the 3rd month of gestation will form the choroidal stroma, while the endothelium lining the blood vessels appear in the choroidal stroma and form choriocapillaries. And by the 4th week, the choriocapillaries will differentiate, and its 2nd month will anastomose with the short ciliary arteries, and at the 8th month, there will be the final arterial circulation is established. So I'm just saying this quickly because it is not of importance. The development of the sclera is ectodermal in origin, so it developed from the neural crystals. However, the posterior margins here and here are developed from the mesoderm. So both ectoderm and mesoderm will share in the same sclera. Started by condensation of mesenchymal cells around the anterior rim of the optic cup, and then they proliferate and secrete collagen and glycosaminoglycans to form the stroma of the sclera. And around the 5th month of gestation, mostly the sclera will be formed. Finally, we reach our final destination, which will be the development of the vascular system of the eye. So basically, we have the artery has three walls. We have the tunica adventitia, which is the most outer layer, the tunica media, which is the smooth muscle layer, and then the most inner layer will be the tunica intima or the endothelium. They have different embryological origins. So the tunica media and the adventitia are derived from the neural crest cells and the ectoderm, while the tunica intima is derived from the endoderm. Let's have an idea about the primary orbital vessels. So while this is the eye and this is the optic nerve, what are the structures or what are the blood vessels that was supplying the orbit during the embryonic life? In the first 8 months of gestation, the orbit and the ocular structures are supplied by three main arteries. Here we have the ventral ophthalmic artery, and B is the dorsal ophthalmic artery, and finally the stapedial artery. This is the stapedial artery, but these undergo some changes. So the ventral artery will disappear. The ventral ophthalmic artery will disappear and it will be no longer present. Only just a small portion which will persist as a long posterior nasal ciliary artery. But where will be the final ophthalmic artery? It will be the dorsal one. So the dorsal one will be the definitive ophthalmic artery. And the stapedial artery will be the middle meningeal artery. So these are the changes that will occur to the primary orbital vessels. For the primary ocular vessels, the developing eye will be supplied by two systems, anterior system and posterior system. The anterior system will be present in the iris and the pupillary membrane and are supplied by branches from the ophthalmic artery, so and anterior ciliary artery and posterior long ciliary arteries. While the posterior systems that supply the posterior segment, which will supply the vitreous and the retina, are derived from the hyaloid system. So the anterior system, as you can see here, the anterior artery system formed by the ophthalmic artery, long and short posterior ciliary arteries, and the anterior ciliary arteries, which are branches from the ophthalmic artery. Here are the anterior ciliary arteries and the posterior ciliary arteries, and here is the ophthalmic artery. These arteries, the anterior ciliary arteries and the long posterior ciliary arteries, will form a major arterial circle at the root of the iris within the ciliary body, as we said before. So you remember the major arterial circle or circus iridis major. This will be responsible for the supplying the anterior segment of the eye. When the pupillary membrane disappear, there will be the remaining the minor arterial circle, which will be supplying the iris only. The posterior arterial system, which will be derived from the hyaloid vessels or hyaloid system. So the hyaloid system will supply the developing globe until the 8th month of gestation. So the hyaloid artery is a primary dorsal ophthalmic, the branch from the primitive dorsal ophthalmic artery, as you can see here. This is the atrophied ventral artery, and this is the dorsal ophthalmic artery that will give an artery that will enter within the optic nerve and enter the eye to supply the posterior segment of the eye. So this is called the hyaloid artery. The hyaloid artery will regress in the vitreous later on and will disappear, leaving the canal of Cloquet, and the part of the hyaloid artery within the optic nerve will persist as a central retinal artery. So again, here is the hyaloid system. The hyaloid system, the artery will enter inside the optic pituitary stalk, enter into the optic stalk from the embryonic fissure, which is a fissure present at the inferior nasal part of the pituitary stalk. This is like an incomplete fold. So if we have a cross section here, we can see this is the optic cup, and this is the embryonic fissure. This is the optic stalk, and there will be an inferior fissure inside the pituitary stalk, making the pituitary stalk incomplete. This pituitary stalk this optic stalk will incorporate the hyaloid artery. So the hyaloid artery will be running inside the optic stalk to reach the eye to supply the posterior segment of the developing eye. Later on, this optic stalk or this embryonic fissure will close, leading to incorporation of the hyaloid arteries inside the inside the optic stalk that will later on form the central retinal artery. So as you can see here, this is the optic stalk, and this is the developing hyaloid artery, and this is the embryonic fissure. The embryonic fissure will later on will close, leading to incorporation of the hyaloid artery within the substance of the nerve fibers of optic nerve. Failure of closure of this embryonic fissure will lead to development of globomas, as we said earlier. So failure of closure of the embryonic fissure will lead to development of different globomas of the iris, choroid, retina, and optic nerve. So here is the branches of the hyaloid artery. The hyaloid artery enter in the optic nerve, supplying the posterior segment, supplying the vitreous, and it will enclose the lens in the forming a bag called the tunica vasculosa lentis. This bag of arteries enclosing the lens are formed of three arterial arcades. So there will be three arterial arcades enclosing the lens, are derived from the hyaloid artery, leading to develop to support the development and nourishment of the developing eye, the posterior segment of the eye from the vitreous toward the retina. This is by the hyaloid system, and this will later on undergo absorption and disappearance. For sometimes there are some congenital anomalies due to a persistent hyaloid system. So the hyaloid system or the hyaloid artery extend from the optic disc toward the enclosing the lens as a tunica vasculosa lentis. Sometimes the proximal part of the hyaloid system will persist and appear as a mobile, non-functioning pterygium-like tissue arise from the optic disc, which doesn't have any significance, no visual significance or clinical significance, but this is just the persistent distal part of the hyaloid artery. Or sometimes the attachment of the hyaloid vessels to the back of the lens capsule will be persistent, and this will be called the Mittendorf dot. So the Mittendorf dot appears as a slightly nasal dot at the posterior surface of the lens due to persistent attachment or the mark of attachment of the hyaloid vessel at the posterior capsule of the lens. So what will be about the retinal circulation? The retinal circulation in the atrophied hyaloid vessels start in the atrophied hyaloid vessels, then retinal vessels emerge from the hyaloid artery to vascularize the retina. So the hyaloid vessels here in the vitreous will atrophy, but within the optic nerve and the retina will form different twigs or multiple twigs to supply the developing retina that will reach the temporal ora serrata it will reach the nasal ora serrata by the 8th month of gestation, while it reaches the temporal ora serrata by 2 months of gestation. So vascularization of the temporal retina will continue after birth. So again, vascularization of the retina will persist after birth in case of temporal ora for 2 months and to reach the temporal ora serrata, while the vascularization of the nasal ora serrata starts or end by the 8th month of gestation. So this is the end of our lecture today. Here is some summary of different ocular structures and their derivatives, and here is a timeline of organogenesis of major events within the eye. Please, if you have any question within the posterior development of the or the embryology of the posterior segment, please let me know in the chat box so that we can repeat it. Any questions? Any concerns regarding the embryology? Okay, I guess that there are no answers. So this will be the end of our embryology lecture and the end of ocular anatomy, which will be the first topic in the exam. I hope that you enjoyed the anatomy and it has become more easy for you to understand and to imagine. So this is just a quick reminder of our next session. It will be the next Friday. This session will be the first part will be the first lecture of optics, which will be the physical optics. And the next lecture will be a revision in the ocular anatomy. So we will solve with past papers questions of from the ICO, and we will this part will be interactive. So we will you will need to submit your answer in the chat box as we will go through the lecture. So after this part, or after the revision of MCQ, we will have our exam as we told you next Saturday. So it will be like today at 6 p.m. GMT. So please try to finish what you have missed in the anatomy, and try to augment your study this week to finalize your study of the anatomy and to have a good score in the anatomy quiz that will be held next Saturday. Thank you very much for your attention, and I hope that you enjoyed the lecture, and see you in the next lecture.